I want you to do something for me. Wherever you are right now, just hold up your hands and take a good look at them.
Yeah, flex your fingers a bit. Really look at your skin.
Right. Now ask yourself, are the hands you are looking at right now the exact same hands you had, say, 10 years ago?
It feels like a trick question, doesn't it? Because intuitively, well, you want to say yes.
You do. I mean, they look like your hands. They have your fingerprints. They carry your scars.
But from a strictly biological and chemical standpoint, the answer is an undeniable paradox.
Exactly. You are the exact same person, but you are made of entirely different stuff. Welcome to the deep dive.
Glad to be here.
The mission today is to explore this concept of what we're calling scientific reincarnation. It's this fascinating, unending cycle of matter that literally makes up the human body.
We've got a great stack of sources to get us there.
We do. We're pulling from a biological explainer on cellular turnover, a briefing from the Pacific Northwest National Laboratory, or PNNL, on biogeochemical transformations, and a really fascinating Reddit thread on the philosophy of biological recycling.
Plus, a cutting-edge forensic taphonomy study from a Canadian research facility, which is just wild.
It really is, because we so often think of our bodies as these permanent, static objects, right? Like a finished sculpture.
Right, but through these sources, we're going to discover that we are actually much more like temporary holding tanks.
And temporary holding tanks, I love that.
Yeah, we're basically just holding on to borrowed elements, constantly trading pieces of ourselves with the universe around us.
Okay, let's unpack this. Because before we can even talk about how our bodies merge with the Earth eventually, we have to grasp that our bodies are in a relentless state of recycling right now.
Oh, absolutely. While we are perfectly alive.
Right, so looking at the biological explainer, the human body contains roughly 30 trillion cells, which is a number I can barely wrap my head around.
It's massive. And out of those 30 trillion, about 330 billion cells die and are replaced by new ones every single day.
Wait, 330 billion every day?
Every single day. That is roughly 1% of your entire physical form, just swapped out for new parts every 24 hours.
That is insane. And the turnover rates for different tissues are completely unsynchronized, too. Like, your stomach lining cells turn over in about 5 days.
Right, because of the harsh stomach acid.
Exactly. While red blood cells max out around 120 days. And the skin cells you were just looking at on your hands, those turn over entirely every 2 to 4 weeks.
It's a continuous rolling renovation.
It's the biological equivalent of the Ship of Theseus paradox.
Oh, yeah, the old philosophy thought experiment.
Right, where if you replace every single wooden plank on a ship over time, is it still the same ship? Apply that to your own biology. If you replace your cells, are you the same you?
It's a wild thing to think about.
But I do want to push back on a popular myth here. We've all heard that trivia fact, that every single cell in the human body is replaced every 7 years. Is there actually scientific consensus on that?
No, actually. That is a highly persistent myth.
Really? I feel like everyone says that.
I know, but the 7 to 10 year time frame is merely the average age of a cell in your body. It is absolutely not a blanket rule.
Oh, interesting.
Yeah, there are cells that stay with you for your entire life. Neurons in your cerebral cortex, the cells in the lens of your eye, certain heart muscle cells, they do not divide and turn over.
So the physical matter constituting those specific cells has literally been with you since birth.
Exactly.
I'm stuck on the logistics of that, though. Because if a cell is microscopic, you can't exactly, cut it open and count tree rings.
Right.
So how are researchers isolating and tracking the age of a single neuron in a living human?
The methodology is actually brilliant. Scientists figured this out using carbon-14 dating, but leveraging a very specific historical anomaly.
Okay, color me intrigued.
So during the 1950s and 60s, there was an unprecedented surge of carbon-14 in the atmosphere due to above-ground nuclear weapons testing.
Wow. Okay, so all that testing just flooded the sky with this isotope.
Exactly. Now, carbon-14 is an unstable isotope, but it gets readily incorporated into atmospheric carbon dioxide, just like normal carbon.
Oh, I see where this is going. So plants photosynthesize that radioactive carbon. Animals eat the plants, and it enters the human food supply.
Precisely. Now, when a new cell is born in your body, it synthesizes new DNA. And in doing so, it locks in the exact ambient ratio of carbon-14 that was present in your environment at that very moment.
That's amazing.
It is, because once that DNA double helix is formed, it does not constantly exchange carbon with the outside world. It is incredibly stable.
So because DNA is stable, it acts like a microscopic time capsule of the atmosphere, at the moment of cell division.
Yes, exactly. By taking tissue samples and measuring those carbon-14 timestamps against the known atmospheric levels of the past 70 years, researchers could prove exactly how old different cells were. That's how we definitively know your cerebral cortex isn't turning over. The carbon signature matches the year you were born, not last Tuesday.
That answers how we track the parts we keep. But it begs a much larger logistical question about the parts we lose.
Okay, lay it on me.
If my body is shedding and rebuilding 330 billion cells every day, the raw materials for those new cells have to come from somewhere, right? And the debris of the old ones has to go somewhere.
If we connect this to the bigger picture, those materials are driven by biogeochemical transformations.
Biogeochemical transformations, that's for the P&L briefing, right?
Right. That is the scientific term for the natural pathways that mediate the flow of fundamental nutrients, things like carbon, nitrogen, phosphorus, and sulfur.
Between living organisms and the abiotic or non-living environment.
Exactly.
So we are essentially looking at the Earth's operating system for elemental recycling.
That's a great way to put it. Consider the phosphorus cycle, for example. Phosphorus is non-negotiable for human life.
Right. It forms the sugar-phosphate backbone of your DNA.
Yep. And it's the key component in ATP, which is the molecule that provides energy to every single cell in your body. But its origin isn't biological.
Wait, really? Where does it come from?
Phosphorus gets released when apatite rocks are weathered down by rain over centuries.
Rocks. We're powered by rock.
Basically, that phosphate washes into the soil, gets absorbed by a plant's root system, you eat the plant, and suddenly a fragmented piece of an ancient mountain is now functioning as the energy currency in your bone marrow.
That is wild. And the carbon cycle operates on the same principle, just via a different medium.
Exactly.
I mean, think about it. The carbon that your body synthesized yesterday to build a brand new skin cell on your hand just a few months ago, that exact carbon atom was likely part of a leaf on an oak tree.
Or just floating in the atmosphere as carbon dioxide gas.
We pull the environment into ourselves, utilize it to maintain our structure for a few weeks or months, and then exhale or excrete it right back out.
You are essentially a localized whirlpool in a much larger global river of matter.
A whirlpool. I like that.
The water is constantly flowing through you, giving you a distinct shape, but the water itself is never the same.
I really appreciate that mechanical analogy. So while we're alive, it's a slow, steady leak of elements back and forth with the Earth.
Right.
But what happens when that system permanently crashes? What happens when a human body dies and 30 trillion cells dump their entire stockpile of borrowed elements into the soil all at once?
To understand that biological shockwave, we have to look to the field of forensic taphonomy.
Taphonomy, that's the study of how organisms decay and become incorporated into the environment.
Exactly. And researchers at the REST facility in Quebec, which is an outdoor taphonomic research space, have been tracking this exact phenomenon.
This is a Canadian study from our source stack.
Yeah, they study the profound biochemical disruption that occurs when a human body decomposes outdoors. It creates what they term a Cadaver Decomposition Island, or a CDI.
A Cadaver Decomposition Island. That sounds intense, because a decomposing human is essentially a massive, concentrated reservoir of proteins and fats.
Oh, absolutely.
It's not just more food for the soil. It's a completely different fuel source. But here's my question. Does this overwhelming influx of nitrogen and carbon spread out and fertilize the surrounding forest, or does it stay contained?
It is shockingly localized.
Really?
Yeah, the taphonomy data shows that despite the tremendous volume of biological matter, the biochemical alterations to the soil are restricted entirely to the topsoil, the A horizon.
Okay, so it stays near the surface.
Right. And laterally, the chemical disruption stops just 20 centimeters away from the edge of the body.
Wait, 20 centimeters? That is barely 8 inches.
I know, it's tiny.
But if you dump a massive amount of liquid and chemical energy onto the ground, physics dictates it should seep further into the ecosystem. Why is there a boundary?
Because the local soil microbiome actively intercepts it.
The bacteria just walls it off.
Basically, the researchers proved this using fluorescence spectroscopy.
Okay, you're going to have to explain that one.
It's a technique where they illuminate soil samples with specific wavelengths of ultraviolet light. Different molecules absorb that light and re-emit it, or fluoresce at different distinct wavelengths.
Okay, so they are effectively looking for the molecular fingerprints of what the bacteria are interacting with.
Exactly. Normally, the organic matter in a forest soil is what we call recalcitrant.
Recalcitrant meaning tough to break down.
Yeah, it's tough, woody, terrestrial material like humic acids from degrading leaf litter. It takes a lot of energy for bacteria to break down.
Right.
But inside the CDI, the spectroscopy revealed a rapid transition to labile compounds.
Labile compound.
Yeah, specifically microbially sourced protein-like substances.
Oh, wow. So the microbes execute a complete metabolic switch.
They do.
They just shut down their slow-burning, wood-digesting factories and fire up rapid response protein-digesting engines to capitalize on the bodily fluids.
I think that's exactly it. They upregulate the specific enzymes needed to digest the energy-rich carbohydrate and protein substrates pouring out of the body.
That's fascinating.
The microbial population explodes and consumes the matter so aggressively that it simply doesn't have the chance to physically spread more than 20 centimeters. They basically build a microscopic wall of consumption.
That perfectly illustrates how dynamic the soil is. But the REST study also highlights that this sequence is not a biological constant.
No, not at all.
It is entirely dictated by the environmental conditions, specifically seasonality.
Seasonality is the driving variable in taphonomic progression.
Right, because in warm summer weather, the rapid metabolic turnover in a Cadaver Decomposition Island completes its primary cycle between 13 and 139 days.
Fast and efficient.
But for bodies placed in the cold autumn months, facing an imminent Canadian winter, the process is delayed to anywhere from 31 to 364 days.
Which is a huge variance.
It is. But let me push back on how we conceptualize that delay, though. Because doesn't freezing weather simply hit the pause button on decomposition? I mean, if you put pizza meat in the freezer, it halts bacterial action. When the ground thaws in the spring, shouldn't the biological trajectory just pick up exactly where it left off?
Here's where it gets really interesting. It is not just a temporal pause. The cold fundamentally alters the biological trajectory.
How so?
Freezing temperatures cause ice crystals to form, right? Which physically rupture bacterial cell walls. It doesn't just put the internal putrefactive bacteria to sleep, it decimates that specific bacterial community.
Oh. So the internal wrecking crew is destroyed before they can even do their job.
Yes. So when spring finally arrives, the decomposer community has to essentially rebuild itself from scratch.
Wow, I hadn't thought of that.
But beyond the bacteria, the physical environment of a spring thaw fundamentally shifts the chemistry. Melting snow saturates the ground, creating extremely wet, low oxygen or anaerobic conditions around the remains.
And how do anaerobic conditions change the breakdown of the tissues?
It triggers a process called saponification.
Saponification. Wait, isn't that how you make soap?
It is. In a forensic context, it leads to the formation of adipocere, which is commonly known as grave wax.
Grave wax. That paints a picture.
Yeah. In low oxygen environments, anaerobic bacteria like certain Clostridium species facilitate the hydrolysis of the body's triglycerides.
Okay.
They break the fats down into free fatty acids, which then bind with calcium and sodium in the surrounding environment.
They literally undergo a chemical reaction that turns the body's fat into soap.
A very hard, waxy soap. And this adipocere forms a protective shell that preserves the underlying tissue and physically locks the nutrients away from the soil microbes.
That is incredible.
Right. Because of this alternative chemical pathway, the disruption to the soil from an overwintered body is drastically delayed, and the ultimate magnitude of the nutrient pulse into the earth is severely muted compared to a summer decomposition.
Wow. Okay, so once those Cadaver Decomposition Islands are finally processed by the soil microbes, whether rapidly in the summer or slowly over a year of freeze and thaw, those human atoms are freed to re-enter the global biogeochemical cycle.
They are back in the global river.
Which brings us to the philosophical yet highly scientific concept of biological reincarnation we touched on at the start.
Yes, the Reddit thread. There was a remarkably insightful discussion online regarding this exact transition.
Right. A user posed a question about the implications of human nervous systems physically breaking down and being recycled into other living creatures.
And the hard science completely validates the premise of that question. The taphonomy data and the biogeochemical models show us the exact mechanism of dispersal.
Right, because a decomposing human leaves behind enriched carbon-13 and nitrogen-15 isotopes in the soil.
Exactly. These heavy isotopes act as stable chemical tracking tags in the environment. We can literally trace them as they are absorbed by the roots of surrounding flora.
And then those plants are subsequently eaten by herbivorous insects and mammals.
Yep.
And those herbivores are consumed by apex predators. Our physical matter systematically disperses outward, woven directly into the broader food web.
The physical boundaries of what you consider to be, you are erased at a molecular level. Your specific atoms become the structural building blocks for entirely new life forms.
There was a mathematical breakdown in that discussion that really crystallizes the scale of this mixing. Statistically speaking, you have about 1 billion atoms in your body right now that were once part of George Washington's body.
1 billion atoms.
1 billion.
The math on that can seem staggering, but due to the sheer volume of atoms in a human body and the thoroughness of atmospheric and aqueous mixing over centuries, it is a statistical certainty.
Because matter is neither created nor destroyed, just endlessly rearranged. So you, the listener, are currently operating a physical body that is the mosaic of recycled parts.
A mosaic, I love that.
You possess carbon atoms in your hands right now that were once integrated into historical figures, part of the cellulose in ancient redwood forests, part of the calcium in Cretaceous dinosaurs, and originally fused together in the nuclear furnaces of long-dead stars.
This raises an important question, though.
Oh?
What does this continuous material recycling mean for our understanding of consciousness?
Oh, man. That is the million-dollar question.
The Reddit discussion inevitably moves toward concepts like Roger Penrose's explorations of panpsychism.
And psychism.
No.
That's the hypothesis that consciousness might be a fundamental, ubiquitous property of matter itself, right?
Exactly, because science can perfectly map the physical elements. We can measure the carbon-13 isotopes transitioning from a human neuron to the soil to the leaf of a plant.
We have the receipts for the atoms.
Right. But philosophy continues to grapple with the experiential aspect. If the precise atomic structures that once facilitated a memory or a thought in your brain eventually assemble to form the sensory organs of a bird,
Right.
How do we contextualize the persistence of experience?
It perfectly bridges the gap between rigid biochemistry and deep existential philosophy. So what does this all mean?
It's a lot to process.
Let's summarize the mechanisms we've explored today. We started with the biological reality that you will systematically replace 330 billion cells in your body today alone.
Just today.
We traced the biogeochemical pathways that supply the phosphorus and carbon required for that continuous turnover. We examined the rigorous science of forensic taphonomy, detailing how our bodies eventually form highly localized cadaver decomposition islands, triggering dramatic metabolic shifts in soil microbiomes.
The microscopic walls of consumption.
Exactly. And finally, we tracked how those atomic components achieve literal scientific reincarnation, dispersing into the global food web.
And understanding the mechanics of this cycle provides us with profound opportunities for the future.
Oh, for sure.
Consider how modern oncology utilizes the mechanics of cellular turnover. Chemotherapy functions by targeting the metabolism of rapidly dividing cells, allowing us to attack a tumor without destroying the surrounding stable tissues.
It exploits the speed of nutrient processing.
Exactly. Now imagine applying that same logic of targeted metabolic manipulation to environmental remediation.
Oh, wow.
Through taphonomy, we now know exactly how specific microbial communities inside a cadaver decomposition island selectively upregulate enzymes to rapidly break down highly complex, concentrated organic matter.
Right, the metabolic switch from recalcitrant to labile.
Right. So if we can isolate the genetics of those specific hyper-aggressive decomposer microbes, could we reverse engineer them?
Oh, I see where you're going with this.
We could theoretically deploy specialized microbial communities designed to metabolize complex environmental pollutants, like synthetic polymers, microplastics, or crude oil spills.
By harnessing the precise mechanics of human decomposition, we might be able to rapidly transform toxic contaminated zones back into thriving, nutrient-rich soil ecosystems.
Exactly. It's a completely new way to look at decay.
Leveraging the biological mechanisms of our own decay to actively engineer the healing of the planet. That is a phenomenal synthesis of the science.
It's all connected.
So the next time you feel static or feel like nothing in your life is changing, just hold up your hands again. Look at your skin. Acknowledge the 330 billion cells you are actively trading with the Earth today.
You're a localized whirlpool.
You are a localized whirlpool in a river of matter. You are an ever-changing masterpiece, participating in an endless ancient cycle. You are never exactly the same person you were yesterday, and that's a beautiful thing. Thanks for joining us on this deep dive.